In this lesson, we learned about vaccines, the immune system's training wheels. Vaccines are not medication, and so we need to be especially sure that they are safe and can genuinely provide protection. Vaccines work by giving the immune system time and opportunity to safely study a modified pathogen, one that structurally resembles the virus or bacterium but is not dangerous. Then, well-trained and perfected T and B cells are stored in our memory, all this without having to suffer disease. Here are some points that can help you review vaccines. Smallpox vaccine was the first and has led to the eradication of this disease. Jenner discovered that inoculation with cowpox protects us against smallpox. There are two types of polio vaccines, IPV and OPV. IPV, the inactivated polio vaccine, is Salk's formaldehyde-killed poliovirus, and OPV is Sabin's live attenuated oral polio vaccine. Attenuation is the result of culturing viruses in suboptimal conditions for many generations. While viruses mutate and adapt to the culture conditions, they lose their ability to cause disease in humans. Subunit vaccines are based on using only fragments of the pathogen. Thus, we can use inactivated bacterial toxins like diphtheria toxoid and tetanus toxoid. Viral subunit vaccines typically contain just the viral spike protein, like in hepatitis B surface antigen and L1 protein from human papillomavirus. These vaccines are especially safe. This is because the viral antigens used for the vaccine are usually produced as recombinant proteins, proteins produced when only the purified gene of the viral antigen is translated in totally safe cells like baker's yeast. Therefore, the vaccines themselves do not contain any viral DNA or RNA, and so aren't infectious at all. Some viruses seem to be resistant to vaccination. An AIDS vaccine is still a real challenge. This is because HIV integrates in our chromosomal DNA, and thus the virus can spread without producing freely circulating virus particles. HIV attacks T cells and thus weakens our immune system. And HIV is constantly changing like a moving target. Commercial vaccines contain adjuvants, chemicals that enhance the immune response towards the vaccine. Typically, adjuvants are aluminum salts in very low concentrations, about 250 micrograms per vaccine. Thimerosal was another ingredient used in vaccines which caused concern. This is because thimerosal contains mercury. However, all the concerns about mercury in vaccines are no longer relevant because thimerosal has been omitted from kids' vaccines since the year 2000. Finally, we reviewed the current vaccination schedule that protects against 13 pathogens. Surprisingly, all these vaccines put together contain fewer antigens than the single shot of smallpox vaccine that has successfully eradicated the disease. Here, in the following concept map, you can see how antigens, pathogens, and vaccines are connected. Pathogens can be viruses and bacteria can present toxins and capsular sugars as antigens. Viruses present proteins as antigens. The antigens can bind B cells. The B cells then produce antibodies which neutralize the pathogens. Activated B cells also produce memory B cells. These antigens are also the basis of vaccines. Vaccines can be live vaccines, such as the case of cowpox vaccine, which protects against smallpox. Killed vaccines are viruses treated with formaldehyde, as in the case of the Salk polio killed vaccine. Attenuated vaccines are viruses that have been treated for many generations in suboptimal conditions. They gain mutations, so they no longer cause disease. The Sabin polio vaccine is an attenuated vaccine. And finally, subunit vaccines present only fragments of the pathogen, typically as recombinant proteins, such as the case of the hepatitis B surface antigen. So, think about it. Vaccines have eradicated disease and saved millions of lives over the years, and will continue to do so for many more years to come.
Cut! And it's a wrap!
Woo!
Yay.